Hello, everybody. In this lecture, we're going to talk about nutrients that microbes require for growth. So let's go get right into it. By the end of this module, you should be able to define the terms essential nutrient and growth factor and describe the energy sources required by phototrophs versus chemotrophs. Alrighty. And so to start out, it's important to know that microbes use nutrients from the environment, and that's how they grow to divide, build their structural components like their cell walls and their enzymes, their cell membranes, and anything else that the microbe needs to make. Now, the vast majority of the dry weight of a cell is going to be carbon, hydrogen, non-gaseous oxygen, and nitrogen. And so three of these, carbon, hydrogen, and oxygen, are going to be commonly found in organic compounds, while nitrogen is also organic as well, but you'll see many different organic compounds that have CH and O. Now, there are two types of organisms that we can think of when we're talking about how organisms get organic carbon. So organisms like us are known as heterotrophs. We require some sort of external source of organic carbon to make our own organic carbon, right? So this can be sugars or fats or proteins. So we're heterotrophs. Many bacteria are heterotrophs. Um, Most fungi are heterotrophs. Pretty much all animals are heterotrophs. And so these are just basically organisms that eat or decompose organic matter to obtain their own organic matter. On the other hand, we have autotrophs. These organisms don't need an external source of organic carbon. They actually convert inorganic carbon into organic carbon. Now, the most common way that you are probably familiar with is through photosynthesis, which is a process of using light to fix carbon dioxide to become sugars, right? And so this little image kind of shows you this kind of intersection here where you have organic compounds that are going to go into your heterotrophs while carbon dioxide and water will go um, into your autotrophs, and you just kind of have this circular connection. Now, without autotrophs fixing inorganic carbon, there would not be organic carbon available for the heterotrophs. And that's an important thing to know. So we've got some things called essential nutrients. These are going to be things that microbes require to build a new cell. So when they're going to begin to divide, they need these specific nutrients to be able to do that. Many of these, especially for your heterotrophs, are going to be found in organic and inorganic compounds that they have in their environment. This is going to be broken up into your two major groups, which are macro and micronutrients. Macronutrients are things like carbon and nitrogen, phosphorus, while your micronutrients are going to usually be used in quite small amounts. These are going to be some of your metals like iron and copper and zinc and magnesium, and some other things as well. And so I mentioned the importance of carbon, hydrogen, and oxygen. You're going to find carbon, hydrogen, and oxygen in pretty much all organic molecules inside the cell. There are a couple examples of organic molecules that don't have oxygen, but they're not usually things that are going to be used to make cellular components. So a big one for carbon is obviously going to be carbohydrates. Here are some hexoses that include glucose, and some riboses, or sorry, some pentoses that include deoxyribose and ribose. Then we also have carbon, hydrogen, oxygen found in our lipids. So here's like a fatty acid chain. We see this long carbon hydrogen chain with the oxygens on the end. And then, of course, we have our amino acids that are going to be used for making proteins. And then our nucleic acids, which have the sugar phosphate backbone, as well as our nitrogenous bases, both of which containing carbon and oxygen and hydrogen. Now, nitrogen is this additional important nutrient. Um, this is going to be included in several different molecules, such as amino sugars. So these are sugars that have been modified with this amine group, with this nitrogen group. These might be used for things like signaling molecules. They might be used for modifying proteins and things like that. You can find nitrogen in fatty acids and that choline at the end of that fatty acid. Of course, we can find them in proteins, right? Because all proteins, all amino acids have a nitrogen in them. That is the amine group. And then the nucleic acids, where we have our nitrogenous bases. So nitrogen is going to be important in all of those. So without nitrogen, we can't build those things. So there are other important elements that aren't carbon, hydrogen, oxygen, nitrogen. They include things like sulfur and phosphorus, and again, some of those metals. Phosphorus is a big one. It's probably one of the most abundant molecules you need outside of carbon, hydrogen, oxygen, and nitrogen. So you can get phosphorus will end up phosphorylating a lot of things. So this can be used for like signaling. So this is an example of a phosphorylated sugar, a sugar phosphate. This is also done during metabolism. So glucose gets converted to glucose 6-phosphate during glycolysis. Obviously, they're a major part of phospholipids. So you've got this phosphate head here. And then you can have phosphorylated amino acids like this phosphothreonine that you can see here. There are other phospho amino acids. The reason this happens is this is sometimes ways that you can modify proteins post-translationally, where you add a phosphate group to them. It can also be used as a signaling molecule for different amino acids. And that phosphate group can alter the way that the amino acids interact with other amino acids, which in turn affects its the protein structure. And of course, we have phosphate in our sugar phosphate backbone of DNA and RNA. Now, most of a cell's nitrogen and phosphorus are extracted from organic nutrients, although some cells get their nitrogen from the atmosphere. And I would actually argue that a significant amount of nitrogen in aquatic environments and in soil environments, or even marine environments, are going to come in these inorganic forms of ammonia and nitrate, right? So ammonia and nitrate are very bioavailable. Of course, as heterotrophs break down organic molecules that have nitrogen in them, that is also going to liberate that nitrogen, usually into some sort of ammonia or nitrate, which can then be taken up by the cell. And so, for example, if you're breaking down a protein and you break off that amine group, you end up with a bit of ammonia. That ammonia can be taken into the cell. So even if it originated from organic matter, it's oftentimes going to end up in inorganic form before it's actually getting into the cell. Now, phosphorus, on the other hand, is kind of interesting because phosphorus is primarily driven by the weathering of rocks, right? So the cycle of phosphorus. And so for microbes in the soil, for example, to get phosphorus, they need to be able to either get it out of decomposing organic matter, or they can get it from just, from as it breaks down in the soil from rocks, as rocks weather, then it releases phosphorus and the cells can bring them up. And I'm not really worried about you guys understanding the nitrogen or the phosphorus cycle per se. I just kind of giving you context for where these things are coming from, like globally. All right. So oftentimes microbes have the ability to build a lot of their own organic molecules that they need for growth, like the 20 amino acids and vitamins and nitrogenous bases. However, sometimes they can't. And when they're unable to synthesize one of these things, that's called an auxotrophy, auxotrophy. And so these auxotrophies occur when you are unable to synthesize a specific growth factor that you need to build a cell, right? So for microbes, this can, I mean, it happens for microbes and for humans, but this can be anything. And so what this means is that certain microbes will not grow in certain environments if those environments don't contain that growth factor. An example of this would be like Neisseria species. So Neisseria gonorrhea and Neisseria meningitidis both fail to be able to produce NAD plus or NADH and hemin. And so they need both of those supplied to be able to grow. Now they can get those during infection, or if we grow them in media, we need to supplement the media with those things for them to be able to grow. Now, there are some organisms that are kind of difficult to grow. They have many auxotrophies. And so if you're trying to grow them in the lab, you have to provide many additional nutrients. These are known as fastidious organisms. And they may need you to provide amino acids or vitamins and so forth and so on. Now, earlier we talked about the different ways of getting carbon into the cell, and that had to do with autotrophy or heterotrophy. Now, we also have two different ways to obtain energy. We have phototrophs. These are organisms that use light energy, and then we have chemotrophs. These are organisms that break down chemical compounds for energy. And so if we look here, we can combine those things where you can have a photoautotroph, which means that they get their energy from sunlight and their carbon from CO2 or an inorganic source. Or you can have a photoheterotroph, where you get your energy from sunlight, but you actually get your carbon from organic matter. Then we have those, the combinations for chemotrophs as well. In your chemoautotroph, you're going to get your um, energy from a chemical source, and you're going to use an inorganic carbon source. So this might be like denitrifying organisms. They often are able to fix CO2 during the process of denitrification. And then we have chemoheterotrophs, which are going to get their energy from breaking down, nutrients, and they're going to get their carbon from organic matter. And so this is going to be like humans, right? Or E. coli, or most of the organisms that cause disease, if not all of them. So with that, we should be able to now define essential nutrients and growth factors, as well as differentiating between the different trophies. That's phototroph, chemotroph, autotroph, and heterotroph. All right, I hope you enjoyed, and talk to you soon.